As we as the topics of the webinars are majorly interconnected to one another, we request you to go to our YouTube channel and watch our YouTube videos based to know about the other topics that we have covered in the last session. Okay. Biochemical tests are the tests we perform on our cultures to identify them based on their reactions to various biochemical compounds. Okay, the organisms produce organic compounds on reaction with the biomolecules, and this results in change of color. The results can range from observation of the growth in the media or up till the change in color due to metabolic reactions. Okay. Like every other procedure in microbiology or anywhere, biochemical tests also have their pros and cons. Okay. Biochemical tests are important because they give both quantitative and qualitative information of our organism. They give us, these are easy to perform in our laboratories and are inexpensive in nature. Or on the other hand, our biochemical tests are very lengthy. Okay. Um, which means that every time we go perform a simple test, there are a lot of steps that we need to follow. It starts from media preparation to inoculation, incubation, and then sometimes you need to even add a certain amount of certain tests need reagents to be added to get a proper result. So these are actually laborious and time consuming, and sometimes there are chances of false positives that can occur due to minor contamination in our test procedures. These tests are extensively used for analysis in healthcare, epidemiology, pharmaceutical, and forensic science studies. Okay, let's go with our first test today. Okay, catalase test. This test is performed to check if our organisms can produce catalase enzyme. We need to know that catalase enzymes help break down hydrogen peroxide into oxygen and water. When in the presence of catalase, we subject our pure culture to hydrogen peroxide, and if we observe quick and brisk effervescence, this during this reaction, it indicates that our organism is catalase positive. And if there is slow or no release of effervescence, that means that our organism are catalase negative. Okay, catalase enzyme is only released by aerobic and facultative anaerobes, and it is completely absent in aerobic microorganisms, and that is because they survive in the absence of oxygen. Okay, catalase can be performed by two methods. One is tube method, another is a slide method. In tube method, we are going to pour 1 to 2 ml of hydrogen peroxide into our test tube, and using a sterile wooden stick or a glass rod, we are going to take a 18 to 24 hour old inoculum of our test organism and mix it with the hydrogen peroxide and observe for immediate bubbling. In slide method, we are going to take our colony, which is like 18 to 24-hour old colony, onto a glass slide, to which we are going to add 3% of our hydrogen peroxide solution. And then we observe for evolution of oxygen bubbles. We conclude that if we see any effervescence during this process, it is called, we can conclude that our organisms are catalase positive. And if we see no bubbles, that means that our organisms are catalase negative. During performing these tests, we need to make sure about these two things. Okay, we need to be careful because you can get false positive reactions if our culture, if we grow our organism on a culture media containing catalase, such as in blood agar, or if we use an ion wire loop while picking up our colony for this reaction. Can anybody tell why we are not supposed to use an ion wire loop for this reaction? Guys,
Please interact, guys.
okay, so nobody knows why it is used, like why it is not used during catalase. Never mind. Okay, I'll let you know why we do not use iron wire. Because iron, when it comes in contact with hydrogen peroxide, without the presence of culture media, also it is going to release of oxygen bubbles are going to happen there. So even if your culture media is not present on the inoculation loop, you will see effervescence, and that is why there are chances you'll get false positive. That is why we don't use iron wire loop. Okay.
Okay. Now we come to oxidase test. Hope you have understand the catalase test. If you have any doubts, you can ask us. Now I'll be explaining you the oxidase test first. So here we will be checking if our I don't know if you are hearing some background noise, something is going on. So if I can't help. So like here we are checking if our microorganism has cytochrome oxidase enzyme, like it produces cytochrome oxidase or indophenol oxidase enzyme, it produces or not. So what is this enzyme? You must have come across the term cytochrome and the terms electron transport chain while you were in your bachelor's or in your master's days, like you studied it in photosynthesis or something. So there cytochrome comes into play, and there are different types of cytochrome. It is a protein, and there are different types of cytochrome A, B, C. So these produce the cytochrome oxidase enzyme, which helps in like in the last step of ETC, if you remember. Like there is oxygen and it uptakes the electron, like it accepts the electron which is given up by NADH. So if you remember that, so in that particular step, cytochrome oxidase helps in shuttling of electrons, from NADH to oxygen and everything. So it helps in that part of the reaction. So here, whichever organisms will be having oxidase, I mean cytochrome oxidase enzyme, are generally because aerobic organisms will only have cytochrome oxidase in them, right? Because oxygen will be the last terminal acceptor only when the organisms are aerobic in nature. So, like this test also tells us if the organism is aerobic or anaerobic. So here what will happen is this reagent N dash N dash tetramethyl P phenylene diamine dihydrochloride. I know the name is too long, but this reagent like helps. This acts as a terminal electron acceptor, and in the case of oxygen, this will accept the electron from NADH, and the color will change from colorless to indophenol blue. So there are several ways by which you can do oxidase test. There is a swab, like we have given a flowchart of all the various types of oxidase, like various methods by which you can do oxidase test. It includes filter paper method, direct plate method, swab method, impregnated oxidase discs or strip method, and test tube method. So all of them use this reagent, and this reagent accepts the electrons when given up by NADH, and then the color is changed. So the end product of this metabolism is either water or hydrogen peroxide. Okay, so which can again be tested by catalase test. So here you have to make sure that you do this test like really quick, because as you know that oxygen is present. I mean, the presence of oxygen can interfere with this test, and atmospheric oxygen is present, and it can give you false result. It can, like, if you just keep your swabs in atmospheric oxygen also, it will change its color eventually. So you have to make it really quick, and you have to make sure that you don't get any false results. And this oxidase discs, as you can see here, this generally have the this reagent in them, and you just have to take one disc, like you don't touch them, you just take it with a sterile forceps, and then you add your culture, pure culture, to it, and if the color changes, it's positive. If the color doesn't change, it's negative. And in this reactions, like you can see, like a reagent is used, which is again Kovacs indole reagent, sorry, Kovacs oxidase reagent, I'm so sorry, Kovacs oxidase reagent you will get. So in that also you get this, I mean the reagent has this reagent, this compound in it, and it changes its color from colorless to blue. I hope the oxidase test is clear to you now. If you have any doubts, you can ask us, or we'll proceed with the coagulase test. Please say yes, no. We are not able to understand your understanding or not. Okay, proceed.
Okay. So next test is coagulase test. Coagulase is an enzyme-like protein that clots plasma by converting fibrinogen to fibrin. This test is majorly done to differentiate between Staphylococcus species. Okay, that is Staphylococcus aureus, which is a coagulase positive organism, and Staphylococcus epidermis and Staphylococcus saprophyticus, which are coagulase negative organisms. Coagulase found in Staphylococcus is of two types. One is bound, another is free coagulase. Bound coagulase is attached to the bacterial cell wall and it enzymatically converts fibrinogen. Sorry for the disturbance. Enzymatically converts fibrinogen to fibrin and causes clumping of plasma, whereas in free coagulase reacts with coagulase reactive factor, which is a plasma factor to produce a thrombin like enzyme that help conversion of fibrinogen to fibrin. Okay.
If you guys don't interact, we are not able to understand if you are getting what we are
Yeah.
saying. So please do interact with us. Please say yes, no. Then only we will get a idea that you are understanding or not, or we will explain it again if you are not understanding. Please make sure that you understand these concepts, because these tests are required all the time. If you go for masters, if you go for PhD and everything, it's very important to understand the principle behind these tests. So make sure that you understand the concepts, and don't memorize it. You just understand what is the reaction taking place, and then you will remember it forever. You will not forget.
Still no response. Okay, so we are going to be positive and say that okay, we are understanding the uh, explanation that we are giving to each test. So we'll proceed with the coagulase test. Okay. So coagulase test, as we discussed, can be conducted by two methods. One is slide test, another is tube test. In slide test, what we do is we take drop of saline on two different slides. Okay, and to each of these drops, we are going to add our colony with the help of either a loop or a wire or a wooden stick, and we make a thick suspension. Now, to either one of these suspensions, we are going to add human or rabbit plasma and mix gently and look for clumping for the next 10 seconds. Okay. We do not add plasma to another suspension because we want to see if our coagulation, if seen, is due to the coagulase enzyme or it is the characteristic of our organism. Okay. In tube test, first what we do is we prepare a dilution of plasma and saline in the ratio 1 is to 10. Okay, we prepare this dilution. Simultaneously, we take three tubes labeled as T, which is test, P, which is positive control, and N, which is negative control. Okay, we are going to pipette zero point 0.5 ml of the dilution that we prepared, the dilution of plasma, into all these three tubes. Now to the tube labeled as T, we are going to add 5 drops of our test organism. Okay, suspension of our test organism to the tube named T. To the tube named P, which is positive control, we are going to add 5 drops of our Staphylococcus aureus culture broth. And to the tube named as N, which is negative control, we are going to add 5 drops of our sterile broth. Okay. Can anybody turn on your mic and tell me why we are using Staphylococcus aureus as our positive control?
Or you can just type it in the chat box if you are reluctant to.
Yeah, okay. If you're nobody knows.
I don't think they're understanding anything.
Yeah. In the previous slide, I just mentioned that Staphylococcus aureus is used as our it is always positive. It's literally written on the first line. Okay. Staphylococcus aureus is standardized organism which gives coagulase positive all the times. Whenever you test it, it's always going to give a positive result. So that is why we use it as a positive control to check if the coagulation has reached up to the mark or not. Okay. So now we are going to mix each of these tubes properly, respectively, and then we are going to incubate these. Okay. Then we are going to examine for coagulation after 1 hour. Okay. After 1 hour, if you still don't see any coagulation, then we are going to examine these tubes at the interval of 30 minutes for the next 6 hours. Okay. We can say our organisms are coagulase positive if our organism produce fibrin clot, no matter what size they are. If there is clot formation, it is coagulase positive, and if you see no clot or no coagulation, that is coagulase negative. Okay. While performing this test, we need to make sure about two things. Okay. One, if we observe clumping in both this slides, in the slide test, if we observe clumping in both the slides, then we it indicates that it is auto-agglutination, which means that our organisms are not showing proper results during slide test. So we have to go ahead and perform this test by tube test method. Okay. Other case is citrate utilizing bacteria. Okay, citrate utilizing bacteria may show false positive by slide test. So in that case also, we are going to go ahead with tube test. Okay, these are the two things we need to remember here. Next is motility test. This is a simple method to test the motility of our organism. Okay, this test is
Can
conducted by
you
two
please
methods.
explain the auto-agglutination again?
Sorry.
Yeah.
Can you please once again explain that auto-agglutination part?
Okay, I will. Okay, so there
Yeah,
are
thank
some
you.
organisms that do not need coagulase enzyme for coagulation. Okay, even subjecting them to any of these enzymes, they can show agglutination. So when we are performing our slide test, we are going to use we are going to add plasma in one, whereas in the other we are not going to add. This is going to show if our organism is acting on the plasma and coagulating, or it generally coagulates even in the absence of um, plasma. Okay. It is kind of confusing, but I'm trying to simplify this to you. Okay. If our one is our what do they say, control. Okay. If our organisms act on the plasma and coagulate, they are coagulase positive. But if they do not act on the plasma, in the second suspension that I had mentioned, we have not added plasma there. But if we still see clumping in that suspension, that means that our organism is not showing coagulation due to the plasma present, not because of the coagulase enzyme. It is showing its agglutination due to its normal metabolism. That is what I call, well, like what we call as auto-agglutination. Even without the presence of release of coagulase enzyme, it is going to show agglutination. Understood?
Okay, thank you. I mean, in the tube or in slide where we do not add the coagulase enzyme, the bacteria is even going to give a positive result just for its normal metabolism, right?
Yeah, it usually
I mean,
doesn't happen
without the
in
presence of
tube test.
sorting factors also.
Sorry?
I mean, without adding the plasma also.
Yeah, without addition of plasma also, some organisms show agglutination.
Okay, okay, understood. Thank you.
Uh, this actually doesn't happen in tube test. If, in tube test, it is pretty
Okay,
clear because we are using only one test, one positive and negative. Usually, this
okay,
auto agglutination is seen in slide test, but it's not like null and void rule that okay, in tube test you won't get auto agglutination. It is usually seen in slide test. Okay. Next
okay, thank
is
you.
motility test. Okay. As we discussed, motility test is a simple method to test if our organisms are motile or not. And this can be done by two methods. One is hanging drop method, other one is motility agar method. So first we'll discuss how what hanging drop method is performed. We are going to take a clean cover slip onto which on all the four corners we are going to apply petroleum jelly, as you can see it in the picture. Okay. At the center, we are going to place a loop full of our microbial suspension, very small quantity. And using a cavity slide, we are going to place it on the cover slip in such a way that the concave depression that you see on the cavity slide is going to cover the cover slip. Okay. Now you invert the slide, the cavity slide, and when you invert this cavity slide, the apparatus should be such that the cover slip should be on top of the cavity slide and our drop should hang from the cover slip. Okay. Now we are going to observe this apparatus under microorganism to observe motility. If here we have to take care of one thing. Okay. If we add a lot of microbial suspension onto our cover slip, it is going to spread throughout the cavity and it's going to attach to the concave depression and we won't be able to see the motility of our organism. So that one thing you have to make sure that you're not going to overload our cover slip with the microbial suspension. Okay. Next is motility agar method. Before we go ahead with the method, we'll study what agar is used here. This agar that we are using in motility agar method is SIM media, which is sulfide indole motility media. As the name suggests, it's a combination differential media and it helps in testing for three different parameters, which are sulfur reduction, indole production, and motility of the organism. Okay. Here, what we do is using a straight needle, we are going to pick a young colony from an agar medium and stab it into our agar culture, agar media that is present in the test tube. We have already discussed how stab culture is done in our previous classes. Okay. Now, this test tube is incubated at 35 to 37 degrees Celsius and we observe motility. We can say our organism is motile if our organism moves throughout the test tube, throughout the media, and making it opaque. Um, and like our organisms don't confine themselves to the stab area. They move throughout the media, making the media little opaque, as you can see on the right side. Okay, our organisms are non-motile if they are confined to the stab line. Okay, this is motility. Next, hydrogen sulfide production test. Okay. Um, Here, this test is to test the production of H2S gas. We need both iron compound and the sulfur compound in this test. Okay. H2S is produced when our bacterial sample reduces the sulfur present in the media and its surroundings. Okay. This breakdown by organisms can be performed either by reduction of sulfur containing amino acids, which are cysteine and methionine, Sorry, there was a slight connection issue. Am I audible now?
Yeah, yeah.
Okay, so I was saying, when anaerobic respiration shuttles electrons from
No, I think you should explain it from the first point. Like you lost connection there.
okay, okay, done.
I think there's some network issue going on.
Yeah, is
Now
it
you're,
okay?
yeah, yeah, it's okay
Is
now.
it okay? Okay, so I'll start from beginning H2S production test if it is okay.
Yeah,
Okay. So to test the production of H2S gas, both our iron compound and sulfur compound is needed. Okay. H2S is produced when our bacterial sample reduces the sulfur present in the media or its surroundings. Okay. This breakdown by organisms can be performed either by reduction of sulfur containing amino acids such as cysteine and methionine, or it can happen through reduction of inorganic sulfur compounds like thiosulfates, sulfates, or sulfides during protein degradation, or when anaerobic respiration shuttles electrons to sulfur instead of oxygen, which means that anaerobic organisms use sulfur as their terminal electron acceptor, unlike aerobic organisms which use oxygen as their terminal electron acceptor at the end of electron transport chain. Okay. As a result, sulfur acts as an electron acceptor here and in the place of oxygen, resulting in the production of H2S gas. Okay. The H2S gas produced post metabolism can be observed when it reacts with the iron compound present in our media to form black precipitate of ferric sulfide. And this indicates the presence of H2S gas. Okay. This procedure can be done using three different types of media, which is SIM media, KIA, or triple sugar iron agar. Okay, we inoculate our organism by the method of stab inoculation, and then we incubate these organisms at 37 degrees Celsius for 24 to 48 hours, and then we observe for black precipitate formation in the media. If we see there is a blackening in the media, that indicates the presence of H2S gas. That means our sulfur was broken down to H2S. Okay, and if we see no blackening in our media, that means that our organisms are H2S negative.